Hang on, I got to accept that it's recording. All right, well, welcome everybody. Good morning, happy December. I'm delighted to introduce Dr. Lisa Bero. Dr. Bero is a chief scientist at the Center for Bioethics and Humanities and professor of medicine and public health at Colorado University Anschutz Medical Center. She's also the senior editor for research integrity at Cochrane. And I was trying to remember how many years I've known Lisa, and I thought it's actually probably better not to think about how many years I've known Lisa. I've known Lisa since we were both working within Cochrane, which was formerly a collaboration. As you might have heard during the chat, Lisa spent several years in Australia, and we are fortunate that she's back on this continent. Though, as you'll know from her location, she's in a different time zone. So a special thank you to her for joining us so early this morning in her time. Lisa is a leader in evidence synthesis, meta research, and studying commercial determinants of health, focusing on tobacco control, pharmaceutical policy, and public health. She's developed and validated qualitative and quantitative methods for assessing bias in the design, conduct, and dissemination of research, and has pioneered the utilization of internal industry documents and data registries to understand corporate tactics and motivations. She has written about measuring problems with research integrity, such as conflicts of interest and spin, testing methods to improve research integrity, such as policy development, and assessing how research is used or cited in policy or media. So with that, I'm delighted to turn it over to Lisa.
Great, thank you, Karen. I'm really looking forward to this presentation, and I'm actually looking forward to talking about one of my new and ongoing projects that I've just started since moving to Sydney. And as my slide shows, I have the Cochrane logo there, and this is really a joint project that very much involves my work as in research integrity at Cochrane. So that's why I'm showing that logo as well. So here are my conflicts of interest. I'm going to move some things around on my screen here. So this work actually is a hangover from one of my grants in Australia from the National Health and Medical Research Council. And I do receive remuneration from Cochrane for my work as a senior editor, which goes directly to the University of Colorado. Now, I'm going to be talking today about problematic studies or studies with serious research integrity problems, and I'll get into what that actually means because it's not very straightforward. But first, I want to give you a couple of examples. And this problem has been around for decades, but it really came to the fore during COVID. And many of you may have heard of this study from a group called Surgisphere. And it was published in May 2020, and it was a very large multi-center observational study of chloroquine treatment in patients hospitalized with COVID-19. And it was published in The Lancet, and the patients were supposedly randomized to chloroquine or other treatments, and it showed that chloroquine was of no benefit. And the Lancet study claimed to have enrolled nearly 96,000 patients with COVID to 671 hospitals around the world. Now, Melissa Davey, who's a journalist in Australia, got interested in this because at the time, and I know because this is the month before I left Australia, there were actually no COVID cases hardly in Australia. And when she looked, the study claimed to have enrolled, I can't remember exactly, but hundreds of patients from Australian hospitals. And so she contacted those hospitals, and they said they had no knowledge of the study. They don't remember participating in it. And certainly they didn't have enough patients to meet this requirement. So basically, this study got further investigated based on Melissa's story, and it was found to be based on fraudulent data. And interestingly, this was fraudulent data from a large secondary database analysis. So it was the Surgisphere was a company with just a handful of employees, and they would evidently sell their data, or they would make publications based on their data. And they refused to hand over this data. The study was ultimately retracted, and other surgisphere studies that were published were also investigated. So that was one example that was really discovered by a journalist. And this is another Australian example, because I know Jennifer Byrne quite well, but this is an interesting story. So Jennifer Byrne is a molecular biologist, and in 2015, she was reading about studies on a pretty obscure gene, and she knew it was obscure because she had actually discovered it and knew that it was a gene of basically no importance. But there was this surge of papers examining this gene, 5 of which were from China at similar times and seemingly unrelated. So she started looking closer at these papers. She worked with people who were image detection specialists and found that some of the images had been faked. And there were other oddities, like the same sequence was used as a positive control in one study as in another study, and the text didn't necessarily match the data. And so there were all these little things that suggested these studies weren't quite right. So she published her findings, contacted the journals, some of which issued a retraction and some of which didn't. But basically, this launched an investigation into these papers that were discovered to be from a paper mill. And a paper mill is basically a group that writes papers that you can buy and then publish under your name. So again, the way Jenny found this was, it was the gene she had discovered that was being reported. So this immediately looked fishy to her. So these are a couple of examples, but problematic research is not rare. So in 2009, there was a global study of science researchers, and it estimated that 14% of their colleagues had fabricated, falsified, or significantly altered their data. One editor of the journal Anesthesia estimated that studies with accessible patient data, which that journal had been asking for routinely, nearly half of those studies contained false data, and 14% of them had so much data that they were viewed as fatally flawed. And then in the Netherlands, where I guess people are more forthcoming, researchers were actually asked about their own behavior very recently. And in this survey of researchers across various fields, 4.3% of them claimed that they had, in the last 3 years, made up data or results. 8.3% had fabricated or falsified data, and 50% said that they had committed at least one questionable research practice in the last 3 years. And that could mean things like not submitting all of your data, or basically altering your data in minor ways. So it's really hard, obviously, to get a prevalence rate for this type of problem, but it does seem to be fairly common. But what is a problematic study? So there's really no agreed terminology or definition. Cochrane has a policy on problematic studies, which I'll describe in a little more detail later. But our definition is any published or unpublished study where there are serious questions about the trustworthiness of the data or the findings, regardless of whether the study has been formally retracted or not. And I think trustworthiness of the data is really key to us. If there's just something about the study that makes you think the data aren't real or they're not believable. So why are problematic studies a problem? Well, first of all, they can create harm to patients. And particularly, my interest is really about their inclusion in systematic reviews. And if studies that have these major research integrity problems are included in systematic reviews, then the conclusion might not be valid, and that can create harm to patients. When these stories come out, like the story of Jenny Byrne, or the story of Surgisphere, it reduces public trust in science. It leads to incorrect decision-making, and it has a downstream influence on research. And as I mentioned, my focus really for this project I'll tell you about has been on systematic reviews and how these studies can influence systematic reviews and what we should do about them. But before we all get really carried away and become research detectives, there's actually some risk of going about trying to identify these problematic studies. So if we identify a problematic study and we exclude it, for example, from a systematic review in error, this can also produce false conclusions. There's an ethical requirement to use the data of research participants. So if real data has been collected, but then it's manipulated in some way, and we never use that data, then that ethical requirement is not fulfilled. And the ethical requirement is really to use all of the data in an appropriate way. Corporate interests often make false accusations to discredit legitimate researchers. So false accusations about fraud or misconduct, for example, the tobacco industry, which, as you mentioned, I'm very familiar with, has a track record of this. They've done this to me when I was at UCSF. And these accusations can damage the reputation of researchers, and they can threaten academic careers. So we really need to be careful that when we're making these research integrity investigations, which is what I call them, that we're doing it in a systematic and justifiable way. So I mentioned Cochrane's policy. This was released in an editorial, and Stephanie Boughton, I want to mention here, is the senior research integrity, the research integrity editor at Cochrane. And she's been doing research integrity work for a major publisher for over a decade, so is very familiar with this kind of thing. And Jack Wilkinson is a statistician in the UK at the University of Manchester, who I've been working with on these research integrity issues. So as I mentioned, Cochrane has a policy. We define what we mean by problematic studies, and then we give some guidance about what to do. And the guidance is very detailed. So this is a guidance for systematic reviewers who are conducting Cochrane reviews. So basically, I'm not going to go through all of this, but we tell them, you know, first you search and see, has the study been retracted? Because as I'm sure you all know, that's not always very obvious. And we have links to that in our database of trials. So if it's been retracted, there are other things you can do, like look for all of the papers from that author. We also give people ways to look for published expressions of concern through PubPeer, for example, or through PubMed. And then we recommend that if a study is identified as problematic, it's excluded from a Cochrane review and put into the awaiting classification category. So that means it's not used in any analysis, and it's awaiting classification until further information is obtained. And we also provide sample letters to right authors for further information, and then to also write editors about our concerns about the paper. The one thing we don't have in our implementation guide is a recommended method for determining if a study is problematic or not. There are lots of methods out there, and we felt that we couldn't really recommend one without further research. So that's what we're doing now. We're doing research into what's the best way to detect these problematic studies. And again, I'm talking about systematic reviewers detecting them. This might also be useful to journal editors and publishers, and I'm involved in a group of journal editors and publishers that are also talking about these tools. But systematic reviewers have kind of a special eye, because they may be looking at all of the studies on a particular topic, so they could see patterns across studies, whereas a journal editor might just get one paper and not see patterns across all the papers. So there's an advantage to how systematic reviewers look at a body of evidence. So folks in Cochrane are using various tools to identify problematic studies and have been publishing these. And I've reviewed all these, and they definitely have their strengths. So people in the pain group have published a study looking at research integrity issues in trials in spinal pain. That includes actually statistical checks on the raw data that they've obtained. And this is a very interesting tool that was used in the Cochrane review on ivermectin. And many studies were actually excluded from the Cochrane review on ivermectin because they were deemed to be untrustworthy and the data could not be verified using this tool. So Cochrane reviewers are doing this, but another problem is we're not publishing these research integrity investigations in any systematic way. So another outcome of our project is to develop a searchable archive of these research integrity investigations so that other systematic reviewers can find them easily, or even other researchers or consumers, anybody out there who wants to look at a particular study could see if it's gone through one of these research integrity investigations. So, as I mentioned, there's a variety of tools that have been proposed to investigate problematic studies. And these vary quite widely from testing for statistical anomalies to searching for inconsistencies in reporting of the data, looking at the text, the phrasing, and the style. And then, as I mentioned, scrutinizing publications across an author or an author group. And that can give you insight into kind of repetitive problems or repetitive research integrity problems, like same images used or same wording used or same patients data used. So what we need, though, is really an easy to use screening method where we can have early warning signs that a study might be problematic, and then in corresponding with the authors, hopefully obtain data and dig deeper to see if we can verify the data. Because not every systematic reviewer is really keen to do such an in-depth investigation of every study they might include in their systematic review, as you can imagine. And the reason we need such a tool is that, typical risk of bias tools that I'm sure you're familiar with, or tools to assess methodological quality, they work on the assumption that the data they're assessing is real data, and that it's it's valid. It's come from real patients, and all the data are there. And so the risk of bias tools actually don't catch these kind of signals that we're looking for when we're trying to detect problematic studies. So in our work, we've done a scoping review to identify these tools. And we've also conducted a qualitative study. And this was led by Lisa Parker, who was formerly a postdoc with me at the University of Sydney, Stephanie, who I mentioned, who's research integrity editor at Cochrane, and Rosa Lawrence, who is a research associate working with me here at Colorado. And we wanted to do a qualitative study of experts because in doing the scoping review, one thing we noticed is that there really seem to be these isolated pockets of people who are working on these kind of research detective issues, and they didn't seem to always be talking to each other. And so we wanted to look broadly. So during 21 and 22, we conducted interviews with 30 participants from 11 countries. And they involve people who are self-proclaimed research detectives or these research fraud scholars, systematic reviewers who have looked into this, many of them from Cochrane, but not all, people in the publishing industry. Almost every publisher has a research integrity unit, and this is one of their issues. And then whistleblowers who have detected big research integrity problems. These are very open-ended interviews like we've done in many of our studies. And we basically ask people, when do you suspect fraud? What are the early warning signs? Kind of what kind of tools do you use? Or what do you look for to detect a problematic study? But we also ask them about definitions and terminology, what types of fraudulent publications they tend to identify, and then, like I said, the screening tools. So the terminology is really problematic, let me say, not to use a pun, but we have a lot of descriptors. These are our descriptors, but they cover a lot of things that our interviewees actually talked about. So let's just take one: inappropriate data analysis. That could include falsification of data, research misconduct, questionable research practices, salami slicing, p-hacking, which is kind of a newer term that actually people in research integrity don't want to perpetuate very much, and then detrimental research practices. So the Office of Research Integrity recommends using this term, detrimental research practices. But as you can see, there's a lot of overlap, and what we were trying to really get at here were what were sort of the big descriptors of the problems that people were looking at. And so we really found that people were talking about altering the data, faking or making up the data, major discrepancies in data, often detected between publications, not adhering to ethical or other research standards, the inappropriate analysis, misleading conclusions, plagiarism or copying someone else's work. It's not always text, publishing their own work multiple times, inappropriate authorships, and I'll give some examples, and admitting conflict of interest declarations, funding sources, or making them up. And then actually, this was kind of a new one to me, fake peer-reviewed or made-up peer review. So where journals actually have published the peer reviewers for a paper and even had fake peer reviews, but they were never done. So that was a new one for me. Sadly, all these other ones were not. They're things I've seen in practice. Basically, on the basis of our interviews, we concluded that a problematic study could kind of be grouped into three types, at least for this population that we were talking to. And these were these kind of tweak data studies, our interviewees called it. So basically where you've got actually real data that's been collected, but they alter the results in some way. And that can include omitting data. Then there's the fake randomized control trial. And I love what one of our interviewees said, that, you know, they were looking into one of these and they actually came to the realization that people would actually make up data. And that was a pretty stunning finding to them. And, you know, some of the examples we heard about, you know, they're just completely fabricated by computer. And now this is getting easier to do because people are even using synthesized trials, you know, at the FDA and for regulatory approval. So the software is out there to make up a trial. So these are either completely made up data or also copied data sets from a completely different study. And I saw an example of that long ago when I was working in tobacco, where someone had done a study on children and they just transplanted the entire data set and said it was conducted in children of a slightly different age, but it was exactly the same data. Then there's the commercial paper mills, and one of our interviewees said this is like organized crime, and it really is, because these are very professional organizations that will basically write papers, often trials, but this is also very common in the basic sciences, and then you can buy the paper and put your name on it. And this is also the groups, the paper mills will also produce the fake peer review for a cost. So as I mentioned, we were aiming for a screening checklist that we could test and validate, and that this would trigger further investigation of studies that were flagged by the screening. So these are some of the things we came up or grouped for the initial screening. So I'll go through examples of each of these, but they go from alerts you look for that are already out there to actually examining the raw data. So by alerts, we mean things like publisher tool alerts. There's a lot of these different tools out there. A lot of journals now link to PubPeer, for example. There's looking for retracted references, and in Cochrane, we've really improved our methods for doing that, and we have a closer partnership with Retraction Watch. There's unusual submission patterns, so that means like the author IP address is the same as the IP address of a recommended peer reviewer. That's also one of the peer review examples. Sorry, I keep using this, but it was so stunning to me, where the authors actually managed to redirect the peer review back to themselves and wrote the peer review. Then there's the plagiarism software, which I'm sure you're all familiar with, and then different data checking software. And one question I often get is like, why can't this all be done automatically? And believe me, I would love it all to be done automatically. And so far, we see parts that can be automated or done with artificial intelligence, but not everything so far. So journal details, this term predatory journal, I'm sure you've heard it, and it's an extremely controversial term. So we actually don't use it in Cochrane very much, but basically they're journals that just seem not to be real. So they actually don't have real editorial boards, or when you look at their submission process, it's kind of fishy, or they don't really have a submission process, or they don't really explain what their peer review is. So basically a fishy kind of journal. And then you can have many of these questionable journals are actually the homes for the paper mills. So if you see a journal which has, if you're looking at the patterns, you'll see a journal that has published lots and lots of papers from paper mills, and they're all very similar. And they also tend to have very, very high submission volumes and publication rates. So author details, and again, we're just looking at the publication now, so we're not digging into the data yet. But something that publishers in journals pick up on are multiple requests for authorship changes. And one thing that readers can pick up on is that there's no academic affiliation of the authors, or it's not a real academic affiliation if you look it up. The name of the corresponding author is, that person is non-existent, so the other authors may exist, but not the one you can contact, so there's actually no way of contacting them. And one of our red flags might be actually just that, not being able to contact the authors to get the data. That's definitely a red flag. And I'm talking about current papers. Of course, this, with older papers, this may happen. Then there's the lack of or strangely worded, reporting requirements or research integrity statements, like no ethics statements or strangely worded ethics statement, and no trial protocol registration, oddly worded acknowledgments or funding statements. And I've seen some very odd funding statements or acknowledgments that basically said, things like they don't acknowledge a funder, but they say that one of the authors went to work for such and such a company right after the paper was published. I mean, really, really strange things. So these are, these again are just tip-offs. They're things that might make you want to investigate the data further. Then there's the submission patterns. Again, publishers look for things like no cover letter or a spike in similar submissions to the journals. But things that reviewers can look for in a published paper are there's multiple similar papers in the same journal. And so they might be on the same topic. They might have the same title with like one word switched. They might have the same methodology, presentation style. So that suggests they're kind of these simulated or created papers. Or there's papers that have different author lists, but again, there's a lot of similarities, and particularly when you're looking at the methods and the data. And a lot of these come, for example, with baseline data. So things are different in the paper, but they're using the same set of baseline data over and over. And current topics that are currently being targeted by paper mills are randomized controlled trials on pretty much any clinical topic, nanotechnology, drug delivery for targeted therapy. So when these topics spike, journals know about them, but we don't necessarily know about them. Then we get to the methods and things to look for. An implausible author team. So this is something, of course, that Melissa picked up with the Surgisphere study, is that the author team didn't involve any of the trial sites or the places where data were being collected. There was just a small group from this company. And so this is something where you actually have to have a little content area. So again, this is why it's hard for AI to do this, because there may be contextual details that are missing, for example. So this can be, a hospital doctor who has no university affiliation, who's authoring a study on molecular biology, a single author conducting a randomized controlled trial, and reporting that the length of participant follow-up is incompatible with the claimed recruitment date and paper submission. So there's a lot of papers that are detected this way, where there's just an implausible enrollment, right? Like they're enrolling more people than actually have the disease in the country or something like that. And these, and this happens. And so these are things where you need some knowledge area and you can pick them up. And then there's, the transparency really around the study methods and the results. So the study may be carried out in just one institution and no data are publicly available with the paper. No, as I mentioned earlier, no details are provided for any way, or they're fake details with how to get the data. And there may be no study checklist. In the style area of looking at a publication, here you don't really need the content expertise as much, and there are some AI tools that can help with this. There's a group of linguists who are looking at, I love this, tortured phrases, and that's text that looks like it might have been copied and passed through several translations to avoid plagiarism detection. And basically, plagiarism detectors can really be gamed, and like the paper mills have absolutely no problem getting around them. And some of the people we interviewed are actually very kind of nervous about developing the kind of tools we're talking about, because they think it will be just a way for the really sophisticated paper mills to get around detection. So that is potentially a risk. Okay, and then, so what we're envisioning is, so we look at the data in the paper first, and if this triggers certain questions, then we would write the authors, that's the process, and then ask for additional data. So this again is looking at just the data in the paper, but some of the things that can, as I mentioned, are just implausible details, implausible recruitment. So, you might have a death date that's prior to a birth date in a paper. The live birth date may be impossibly close to the recruitment of non-pregnant participants. This has really been detected a lot, these sort of date issues in papers around neonates and pregnant women. And then you may see that, a very unusually high proportion of participant newborns are of one sex or the other. Female participants maybe in a study of male cancer, or vice versa. The study of some uterine cancer may include women with hysterectomies, and there, may be a fairly large number of them. So there's all sorts of strange things you can pick up in these papers. And then you can look at actually the numbers, and you may see things like identical means and standard deviations in the study and control groups. And again, when looking across papers, what you often see are identical control groups as a tip-off. And then in the results, you can also see outliers and implausible results. And the thing there is there are statistical programs. And as I mentioned, Jack Wilkinson is an expert on this. I am not. But once you get the raw data, you can actually look at some of these statistical anomalies, but you can detect the outliers or just these implausible data by looking at the paper itself. As you can imagine, this takes time. And so this is why we're actually trying to develop a streamlined tool that will trigger a more in-depth investigation. And so that this second pass screen, excuse me, so say you've looked at one paper and a few of these red flags are raised. So in this second pass screen, you may want to look at other publications from the same authors, because this kind of repetition or reuse of data might become very apparent. And then you might want to dig into the peer reviewers, something I would never have done before. You might want to actually check back with the group where the ethics registration is registered, or where the protocol is registered. I have actually gone to look for a number of protocols and never found them. So the paper said the protocol is registered at blah, 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 and then you go there and it's not. And then, of course, ask the authors for raw data. And so in Cochrane, we have a model letter that we use to write the authors to say, we've identified these questions, and it's a very neutrally, worded letter. These are the questions. And then at the end, we would ask for the raw data. And again, if we don't get that, that's sort of a tip off. And then you can actually go to experts to help you with some of the analyses of the raw data, which is, for example, using some of these statistical tests. One is called the GRIM test, or using expert image review. There's a whole little cottage industry in research integrity of people who look at images and do these statistical tests for people. So you send them your image or your data, and not your data, somebody else's data, and they'll run these tests for you. And I remember, I think, Karen, you were asking me, are there really research integrity experts out there? And they are. I mean, there are people who do this for a living, and they'll be called a research integrity consultant or something like that. So the end of our qualitative study was to say, okay, there's a lot of options out there. There's a lot of things that people are looking for, but what would be useful to systematic reviewers, and how can we really pare this down and test it for a tool for systematic reviewers? So Jack and Stephanie and I wrote a grant to NIHR, which was funded, and it's to develop an evaluated tool to detect basically problematic randomized controlled trials. It's called the Inspect SR project and tool. And the aims of the project, which we're about 7 months into now, are to supplement the existing scoping review we've done and qualitative study with a review of the gray literature and another survey of experts. And here we're really going to be focusing on kind of publishers and research integrity professionals, because one thing we discovered in our interviews, which I found quite ironic, was a lot of these research integrity investigators have developed their sort of own tools or their own software to check these things, but they actually don't share them with anybody. They keep them to themselves. So there's stuff out there that is obviously not published, and we're trying to see, what it is. And then we're going to evaluate the impact and the feasibility of the method by retrospectively applying it to a large sample of published systematic reviews. And in this case, we're concentrating on Cochrane reviews. And so the aim one, we're going to have a kind of a large number of items that we're going to check. And in aim two, we're also going to have a fairly large number of items that we're going to check and apply to the actual trials from the systematic reviews. Then in aim three, we're going to use a Delphi process to whittle down this large number of items. And so basically what we're going to feed into the Delphi process is not only the results of the survey, but basically how this actually worked in practice when applying it to the randomized trials. And then once we have that whittled down group of items from the Delphi process, we want to test it again on systematic reviews. And we want to look at the impact on the systematic review conclusions and results. And so how this would work is we have a draft tool. We apply it to the trials in a Cochrane systematic review. If any of those trials are identified as problematic, we then exclude them from the Cochrane review And we see how this changes the meta-analysis, if there is one, so the number of studies, the point estimate, the variation around the point estimate. But then we're also going to look at how exclusion of these might change the grade evaluation of the conclusion, which may include more studies than were just included in the meta-analysis. But kind of the overall goal is to come up with an applicable, and I think that's the hardest part, and valid way of identifying these studies so that we keep them out of systematic reviews. And we think this is really important because there's data, actually, Marcella Salandra recently published an editorial in Cochrane, which I don't have in front of me here, but she did a really interesting paper that showed that when studies in Cochrane reviews were rated as high risk of bias, their citation rate went down over the years. So just identifying them as high risk of bias seemed to prevent them from being used more in subsequent literature. And that's kind of what we want to see with these problematic studies. We would like to stamp them, out of the literature or see them used less. So right now we've got 106 items at this early stage of the Inspect SR tool. And you can see that they fall into different categories: inspecting the results, the research team, the conduct, governance and transparency, text and publication details, and then on to the individual patient data. So we hope at the end of our Delphi, we have many fewer items here. And some of them actually are probably duplicative, but we wanted to start with the biggest set we could get to put into our survey and our applicability test. So that's where we're at now. And I've been writing a lot about this lately. And one question I get is, well, why are you bothering to identify these for inclusion in systematic reviews? Wouldn't it be better to actually just prevent them from being published in the first place? And I totally agree that is a way to go, but it involves a lot of people. And what I have control over right now are systematic reviews and this aspect of methods in systematic reviews. And so that's a good place for us to start. But I do believe that we really need to start as far back as the academic incentives that do things like support these paper mills so that people can get those publications on their CV. We have to get journals and editors involved. And I have to say, publishers are very involved. They work more behind the scenes, is is kind of my perception so far of this. But, it's very embarrassing to them when this happens or when they're caught in a paper mill, for example. And so they really are doing a lot. But I think the big problem is people aren't currently sharing information about how they do this. So one thing, as I mentioned with our tool, once we have it, we'll share it with everybody. And we will also publish all of the research and integrity investigations in a searchable format so that anybody could find them and not have to repeat all that work of doing the research integrity investigation. And systematic reviews are really a good place to start because, as I mentioned, people look across a whole topic, not just at one study at a time, like a publisher or publishers do see across, but like a journal editor would. But I'm not minimizing the problem. It's going to take a lot of folks working together to get rid of it. But I hope we can all do that. And I think that's it for me. So I'm happy to open it up to questions or comments while I have a drink of water here. Thank you.
Thank you so much, Lisa. Please either put in chat or unmute yourself if you have a question. I have actually a number of questions, so I'm just going to ask you the first question, if I may. So first of all, this is scary. It's scary because you think about the reviews that you've done and whether the conclusions are true, right? If you have some problematic studies in there. But it's also scary as a systematic reviewer thinking, how am I going to do yet another task? I'm already, screening thousands of citations, doing data extraction, risk of bias. how do you envision this working in with a typical systematic review process? At what point and how would this be implemented?
Yeah, I mean, well, you hit the nail on the head, and that's why we really want to try to develop a sort of a red flag screening tool, because we do see that it's going to be impractical, for example, to look at the IPD data for all studies. And we see this happening at the stage when you're screening studies for inclusion, because you don't want to do any, so once a study is tagged for inclusion, meets the inclusion criteria, I should say, then we think this is the point at which one would look for these research integrity problems. And in a way that saves time, because then if it's parked in awaiting studies or it's excluded, then you've not done the risk of bias evaluation or any of that. We think that we'll have to target certain areas. The trouble is, I really don't know. The more I learn about the prevalence of this, it seems widespread and not really in specific areas. I mean, I know folks in Cochrane have, told me, oh, it's only in the pregnancy intervention area, or oh, it's only in anesthesiology, or, it's in these particular areas. But I don't see that. So we need to do some, and by that, I mean, like the research integrity teams need to do some preliminary work to figure out if we need to target sort of certain topic areas. And I think that we could get, our goal is to really get a screening tool that, would take less than 20 minutes to look at, per paper.
Mhm.
And if that raises red flags, then there would be further time. But yeah, that's what we need to do. And in Cochrane, I mean, I can't vouch for all systematic reviewers, but in Cochrane, we do have a research integrity team of 3 people, and we do offer support in terms of when a study appears to be questionable, we'll then look at the study and apply some additional checks. We provide the model letters for contacting the authors and contacting the editors, and we'll do that follow-up. So we do have, and anybody can look at our implementation guide, some tools there.
Thank you. Jody?
Thank you. This was very interesting. So a slightly different topic. So I'll be participating in a webinar this Friday for Clinical Research Forum. And the topic is, and very relevant to systematic reviews, the lack of description of data in those big data sources like Optum or Trinetics, which purchase electronic health record data or claims data. And so that there's overlapping populations in all of these big observational studies, and you can't know. And I'm a deputy editor at Annals of Internal Medicine, and we know it's an issue, and there is no solution right now. Do you have thoughts on that?
Well, I do, because we want to move on and are currently writing a grant that Karen is involved in to actually modify our tool for observational data. And we're not sure how that could be done, but the issue definitely arose during our interview studies, because they covered people who worked across, everything from bench science to epidemiology. And so we're focusing on trials now, because that's, they're most prevalent in Cochrane, but even Cochrane now includes, some of these databases. So yeah, we need to work on that too. I don't have an answer yet, but I think we've recognized it as a problem, although the prevalence hasn't, really been written about as much, as far as I know.
No, I don't think you would even know the prevalence.
And another problem is, some of these algorithms that are used to analyze the data, they're secret, right? So it's very hard to even know what people are doing. So I think we have to also get over a transparency barrier before we can, dig into some of this. And that's one of the big, things that's happened, as you know, and it's taken decades with trials, is there's a lot more transparency. We can at least get the data. So that's really the first step.
Mm-hmm. Yeah, thanks.
So I have another general question, and I do see a couple in the chat as well, but you mentioned something quite different than the other tools I've tried, and that is having a registry of problematic trials, essentially, right? Or trials that have gone through this screening. Given the harm it could do to individual researchers or institutions even, how, as a systematic reviewer, that also is scary to me. If I'm going to flag something as problematic and it's going to get posted somewhere publicly as being problematic, what are my legal or other risks in doing that?
Yeah, and that's something we've looked at in Cochrane, because right now we're investigating sort of two relationships. One is with PubPeer, and the other is with Open Science Framework. And Open Science Framework has a lot of advantages in terms of searchability and archiving and giving them a DOI. But both of them have the advantage in that they do screen what's posted for, basically when any open access publication screens for, a libel and what is it? Oh gosh, I'm blanking on the word. Libel, basically. So there is a legal screen before they can be posted. But the other, and I think more important protection is that if people are using this tool, that will legitimize it, because they can always say, well, we're just using this tool and we answered this question, so, such and such. And they also have to provide a justification in the tool. I didn't go into that for any of the problems they identify, like, where in the paper is, the baseline data repeated or whatever. So there is a justification there. And we always contact the authors first before a research integrity investigation is considered complete. So the other thing we would file with the research integrity investigation is that correspondence with the author. Did they respond? What did they say? And then the research integrity investigation at that point, after the response from the author, would be published. So I actually don't see any problems to this, because I'm comparing it to kind of what I've seen now posted occasionally or published that doesn't rely on any sort of screening tool, and that doesn't really go through these kind of checks.
Yeah, it's good. The tool kind of gives you a cover. I know that I was involved in a Cochrane review, where we had a suspicious trial, and at that time it was like, well, what do we do?
Yeah,
yeah.
And that also, as I mentioned, we now have the problematic studies policy. And so one thing, those papers I showed you from the pain group and the ivermectin team, they actually state in them that they followed our policy for problematic studies, because we actually recommend, that, based on your screening, if you think it's problematic, put it in awaiting studies, contact the author, we recommend a certain process.
Yeah. So a couple of questions in chat. So one is, do you think that do you see Inspect SR being integrated within Covidence or other sorts of tools like that?
Oh, wow. That would be great. Yeah. I don't know. I mean, we have to see. Yeah, we have to see. I mean, ideally, we would definitely want it integrated into that kind of software to make it easier to use. Not all the questions are yes, no questions, but I don't think that's a problem. We can have sort of text answers in there. So that would be ideal. And that would be kind of the right place to do it.
Right. And yeah, I mean, there are other tools like Distiller or Pico Portal that you could upload the template into and use. Yeah.
Definitely. Yeah. Yeah.
And then an early question was about plagiarism detection websites. If you have any suggestions for particularly good ones.
Oh, I don't. I have a colleague here in Colorado, actually, Daniel Acuna. If you want to email me, I could forward it to him, because he's actually an AI specialist person who's been looking at plagiarism detection. And I know which ones he thinks are quite bad. Well, I don't remember them, or I would tell you, but he might have some that he thinks are good as well.
Yeah. we're just about out of time, but that reminds me that I think that ironically, I think most Cochrane reviews would get flagged for plagiarism.
Oh, that happened.
Right, because the methods, I mean, how many different ways can you say the method? So, I mean, they would get flagged, wouldn't they?
No, they were when when these started coming out, and then we got contacted. I think on the board, or I was on some big committee at that time, and all of our reviews were getting flagged as plagiarized. And we actually worked with those software developers to get around that problem. But yeah, you're right. There are only so many, and we use all those standard headings too, and they would include headings. Yeah,
right. And same author groups on multiple reviews. Yeah. All right.
Yeah.
Thank you.
So it is a scary problem. I mean, and I'm doing this because I so believe in systematic reviews, but I think this is a huge threat, and people can use this argument against them. So it is important. Anyway, I'll be quiet.
No, thank you so much, Lisa, for sharing this really important work. Really appreciate you, especially getting up so early to join us. Very much appreciate.
Well, I'm happy to stay in touch.
Glad we're working together again. That's great.
It is great. And thank you so much for the invitation. Great to see you all.
Thank you.
Take care, everyone.
Bye.